Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Tarek O'Dali from Business Review, and I will be your host. It is our pleasure to have Capsugel with us today. He'll be presenting this webinar titled Spray-Dried Biologics, Formulation and Process Considerations. Today's guest speaker is Michael Burke, Senior Scientist, Product Development. I'd like to welcome you all to our webinar platform on 24. You will notice this webinar is fully browser-based, so if you disconnect for any reason, please click on the link you received via email to rejoin the session. In order to ask us questions, you can send them in at any time via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. And we'll try to allocate some time at the end of the session to address any questions or thoughts you may have. Please use the yellow Help widget if you do require any technical assistance, and you can move, resize, or maximize any of the windows in front of you to get a better view of the slides. But now that having been said, please allow me to welcome today's speaker. Michael, over to you.
All right, well, thank you, Tarek, for the introduction, and thank you, everyone, for joining today's webinar. During the webinar, we'll be addressing spray-dried biologic, and specifically be looking at formulation and process considerations in regards to product development. And I really hope that from the information provided in today's webinar that it'll be educational and sparks additional discussion. We do have quite a bit actually kind of throughout the webinar, so we'll get right into it. So today's webinar is kind of structured as a technology discussion. Will last for approximately 45 minutes. And we'll be addressing the topics that you can see listed on the slide. So those topics really are, why would we spray dry? An actual spray drying overview, process-wise. General approach, which includes establishing a problem statement and a goal, and then it's biopharmaceutical applicability. And then when looking at formulation process options for spray drying, we have a couple of different important factors that we'll need to consider. What is the actual target product profile, typical excipients and rationale that are included, formulation impact on the process itself, how to scale up the process, and then the corresponding analytical, which we all know is important for understanding the products that we create. We'll move then to a brief case study that looks at spray-dried monoclonal antibodies. And lastly, we'll conclude by just tying everything together. And again, hopefully by the end of the talk, you'll have a better understanding of our general approach for spray drying. And you'll see that what we'll take through this is more of a holistic approach, and understanding each component will be important for product development and design. Now, before diving into the specific details of the webinar, I just wanted to touch base on the breadth of Capsugel's technology platform. You kind of see in the table itself here, we're capable of addressing a number of different problem statements, which includes bioavailability enhancement, targeted release, capsule technologies, and then specialized applications. This presentation itself, of course, is focused more on spray-dried biologics. I've highlighted a couple of different of these subtopics here that will kind of tie into the presentation. So, of course, the spray-dried dispersion, enteric coating of capsules or mini-tableting of dosage forms, how to put those mini-tableted materials into a specific capsule itself to either get for a immediate or delayed release, and then focusing more, again, on the bioformulation aspect of spray drying. Again, in each case, what we'll see is that these are individual platforms. However, throughout the product development life cycle, everything's intertwined and should be under consideration. And I hope you can kind of see that connection as we go through each one of the slides and the topics. So also, in addition to the technology platforms, of course, Capsugel itself has a variety of different manufacturing facilities across the United States and Europe. Each one of these have kind of a specific expertise, and just kind of to orient everybody, myself, I'm located in Bend, Oregon, which is the western portion of the United States. We focus on actually enabling spray drying processes. So our site contains all the necessary manufacturing and analytical equipment to support anything from general research and development to feasibility and clinical applications, and then all the way through commercial manufacturing. Okay, now with that kind of put aside, we'll kind of move now into today's topic and its applications. So as we all know, biotherapeutic agents and formulations are normally delivered in two different ways. That is either a liquid or a solid. Now the liquid is more of the simple approach, and everybody has kind of worked with in the past. Water is a primary matrix. You're including sugars, surfactants, salts, and these are kind of added to support protein stability, prevent surface interactions when you're actually mixing within the air-liquid interface. Formulations can be optimized by just directly adding excipients. They can either then be sterile filtered or diafiltrated for processes downstream. They do usually require for cold storage because you have kind of a mobile protein in this water matrix. As I said before, the approach can be usually quite simple. You can do appropriate buffered screening, which can be vast. And really the main issue around is long-term stability, a storage burden because you're storing all these large amounts of volume of liquids, corresponding patient compliance in a lot of situations because you're delivering these liquid formulations as a kind of IV or sub-Q, and then limits yourself to specific delivery modalities. Now for the solid form, this is kind of the second way of doing biotherapeutic formulations. The technology itself is usually focused on either lyophilization or, in this case, spray drying, which we'll talk about during today's webinar. Again, usually conversion of the solid form is used an alternative, and liquid-based formulations have complications or specific needs. And lyophilization itself is sort of considered as the gold standard. It's been around for many years. A lot of folks have used this kind of in either the small-scale lab or large-scale production. The process has been commoditized over the years. You can go purchase a lyophilizer. But there's usually a lot of issues with scalability because you're kind of limited to that footprint of that lyophilizer and the actual volumes you can work with. Now what we're going to focus on is spray drying. So spray drying itself allows for particle engineering to enable different compositions, enhance stability, and different delivery mechanisms. We can decrease sample footprint because we're introducing now a density component. We control how those particles are formed. We are using sugar as really the primary matrix in this case, and we'll talk about that more through the slides. And also you're adding things like surfactants, polymers, amino acids, salts, a variety of different things that can help stabilize the protein, while also, because we're converting that liquid into a solid during the spray drying process, protect it during that air-liquid interface. The water is usually a constituent part of the process as well because you're processing materials using a solution that's containing water. And it ends up being around 1 to 7%. Of course, that is tunable through the process. And just on the right side of this slide here, just the bottom part, is kind of our holistic approach for spray drying. We'll move into discussing about that just a little bit. Wanted to start off by really just talking about why is spray drying itself appropriate for biopharmaceuticals. Now, there's a reason for this, and it's really based on the fact that we're utilizing an evaporative cooling step. And what that does is really you're processing a material from the liquid form, and it's heating up to where that evaporation point is within the spray dryer. And again, for a wet bulb temperature of water, it's around 40 degrees Celsius. And then the drying temperatures themselves kind of allow for a mild exposure with a really short residence time. And in general, so the fast drying is kind of noted here on this slide. Solidification is a matter of milliseconds as we process through the spray dryer. And each one of the properties within the spray dryer can be tuned to kind of meet whatever product you need or product profile you need. And that's really around optimizing the drying conditions, making sure as material goes and evaporates, we have the right product. And again, this is kind of amenable to different variety of dosage forms. Now the product form itself, when spray drying, is primarily amorphous when coming out of a water matrix for a biotherapeutic. However, you can use the process to force crystallize materials, and that's usually the function of the excipients to help kind of give different powder properties. And we'll talk about that further within the webinar. And again, spray drying itself does have commercial viability. It's continuous and scalable. And I also wanted to note one thing here prior to moving on to the next couple slides is the graph on the bottom right here. It can kind of give you a visual representation of what this means in the evaporative cooling step. And so, of course, we have a solution that's typically processed at room temperature, and then as that droplet is atomized and pushed in the spray dryer, it's heating up within the confines of the dryer. Now it has still water kind of associated with the material. As you then evaporate off, you go up and you're heating up to what ends up being the outlet temperature of the spray dryer. And again, this is just for an introduction. We'll talk more in detail in the upcoming slides. And ultimately, in the end, you'll have a spray drying process that gives you a homogeneous particle that contains the biopharmaceutical components along with the excipients needed for stability. So today's presentation focused really on spray drying. I wanted to start with just the general fundamentals around the process itself. You know, we always state this spray drying as something that is certainly an enabling process, or it can be even a replacing technology if you're using lyophilization. You're looking for larger batch sizes or things like that. But spray drying is fed into kind of four main operations. We have a solution feed, which is noted here as the spray solution. We have an atomization process, which is the solution being fed into an atomizer, which is mixed in with a drying nitrogen gas. That's setting up the thermal profile for the spray dryer. And we have the drying chamber itself, is where the drying is occurring. So we're getting the evaporation of the solvent and then condensing the individual solids within the solution into a particle, and ultimately a collection. Each one of these components are somewhat tunable and screened through early feasibility studies. And they have their own inputs, controls, and outputs. And in the end, the goal is implementing a process to enable a control of particle formation and corresponding engineering. And again, just to state the fact that we have processing solutions, and the solutions can be a variety of different solid contents, but you're just using the drying gas itself to evaporate off that water. The spray drying biologics themselves really has a lot of precedence within the literature. There's a number of academics and other pharmaceutical companies that will look into using this technology. Of course, we've had a lot of experience over the years doing this. Spray drying itself is rapid, robust, mild, and atomization itself can be tailored based on need. So I wanted to dive real into the physical situation of spray drying itself. So again, as I stated before, we have a liquid feed solution. The feed, which is controlled, is atomized using the drying gas, and again, typically a nitrogen source. When this mixture occurs, you form a droplet, and this does introduce the air-liquid interface where a hot gas will contact that surface of the droplet, and evaporation begins, and the droplet skins. So the droplet continues to heat, and the particle morphology begins to form, and the morphology is dependent upon the solute, the solvent selection, and specifically diffusion of the solvent from the materials within the solid content. Kind of see that model there in the left-hand side of the slide as the drying gas goes to the outer portion of the droplet. And then again, utilizing this kind of general holistic model of spray drying itself, you have the individual components that you're worried about when spray drying. So you have typically an aqueous solution that have kind of water-soluble excipients, a variety of different actives that are associated with this. The atomization itself is putting on a little bit of stress, shear stress. There is a selection in the nozzle design, nozzle parameters. You vary the feed rate itself. That can kind of impact overall throughput and also stresses within the particles or materials within the solution itself. The drying conditions will define morphology and the kinetics and thermodynamics. And ultimately the collection, which is based on the density in the cyclone design. And later in the presentation, we will discuss how the selected formulations can impact the process inputs and how we design these to ensure ideal particle properties. The spray drying process development itself, too, there's a couple different offline tools here we'll use to track how we're manufacturing materials. And these kind of are listed on this slide here. You know, doing nozzle characterization on what we use as a nozzle test stand you can look and see how the individual droplets are forming and what sizes they have, what kind of spray plume are we getting, are we going to get a better distribution of particles with it dryer, and then a better drying profile. And then you have corresponding actual collection evaluations. We've done some CFD modeling to make sure that we're getting physical separation of the dense particles and the actual gas itself as a bypass. Understanding kind of what is our thermodynamic operating space. So at what point will we put the protein or the macromolecule in a region where there be instabilities that can be tracked. After collecting materials, you can do studies to look at what is your thermal constraints using a hot bench experiment. This is simply adding powder to a thermal gradient. Look and see how the powder itself will either discolor or we can track impurities. Again, it gives us a real good idea of how the process itself should be tuned to give you the right particles and ultimate products for use. In a lot of situations, too, we're working with hygroscopic materials, and so the water uptake potential, as usually measured by dynamic vapor sorption, can be assessed and tracked to ensure that, when we expose these things to elevated humidities, that we're not going to get secondary impacts and instabilities, and then also because there's a residual water around, that has impact overall potency. Again, just to kind of reiterate, the spray drying process itself is highly tunable, and it should be tracked and broken up into those couple different categories that we just discussed. So the first thing we would do when we have an actual product and we want to develop a formulation of process is to identify a problem statement. By identifying the problem statement front side, we can rationalize which excipients, what is our process constraints, things like that. Kind of listed on this slide here are the four kind of primary problem statements that we encounter. So it could be simply a bulk dry powder formulation. So you have a material, you're looking for just having stability in the dry form. You want to eliminate cold state storage by using the dry form and providing long-term stability. Pretty straightforward in that case. Maybe it's just alternative for lyophilization because of the fact that you have limitations with that process. So spray drying is going to allow for continuous, robust, and scalable processes for large batches. We can go from early feasibility, which would be minimal drug developments, and be able to translate all the way up through. Maybe then there's different excipient profiles that you need for stabilization of the molecule. With that, lyophilization has its set of excipients that it can use for the cake formation. The spray drying opens up a lot more avenues for inclusion to design the particle. In a lot of situations themselves, too, we're looking at high concentration reconstitution. So you have a limited solubility of the active within the liquid formulation, but by spray drying and adding functional excipients, you can actually increase the apparent resuspension concentration and quality. Again, this is all about a tailored formulation to meet your goal. And again, ultimately, the particle engineering component of spray drying is critical. We can really control around densities, flowability, so how's it going to handle. We can have targeted delivery, specifically pulmonary or nasal or oral delivery. And we'll touch on this a little bit more when we go into more formulation specifics. But we can also have specific product morphologies as well that will either impact your reconstitution, your filling, and your flowability. So with these four primary problem statements, we've also applied this to a number of different biologics themselves, and kind of listed on the right-hand side of this slide now. We've done this, or applied this approach to peptides, proteins, monoclonal antibodies, different vaccines, whole cells in general as well. So in each one of these approaches, understanding the molecule, combining these together are really critical to success in the end. So now we've established a problem statement. Now what we need to also understand is what is the actual target product profile. And to get to this, and before understanding a formulation or process, we need to think about the molecule itself. So, we kind of have this model listed here on the right-hand side of your slide here. And this is how we tie into things and think through things during the design phase. We have a definition in a compound and feeds into our formulation and process, using the characteristics of the molecule, feed that into the spray drying process, getting our downstream characterization, and then our ultimate dosage form. So, what are those criteria that we consider right away? We have molecule type and weight, of course. What's the isoelectric points? What's the preferred pH of the molecule itself? Where will we have instabilities there? How hydrophilic or hydrophobic is it? What is our overall solubility? Because that'll define what our initial solution or throughput may be, and also may impact what excipients we would add. What's the overall dose and concentration we need if there is a defined delivery profile? Are we looking at something that's acute or chronic delivery itself? And then also, is it something that's actual local delivery? So specifically thinking of things like pulmonary delivery, where we want a molecule to stay in the lung, or again, maybe even in the oral form, you're looking for receptors in the epithelial layer of the gut. Or is it systemic? So we need rapid kind of dissolution and transfer. So again, that defines what kind of excipients we would add, how we would think about the active, what's the right way to go forward. So let's kind of say there's additional qualities as well. So that delivery mechanism. So are we looking at something that's IV, sub-Q, inhalation, intranasal, oral? Knowing what that ultimate delivery mechanism is, we would have to then define particle size and overall dissolution properties, including as well, so kind of reconstitution profile we need to rapid reconstitution, high concentration reconstitution. What's the overall molecular stability? What kind of temperature profiles are these? What kind of excipient incompatibilities do we have? And in the end, because we're converting something to the dry powder state, we care about what is the actual glass transition temperature. So what point do we see mobility as a function of temperature, and corresponding decomposition and melting temperature. So again, taking in all of these kind of early understandings of the molecule, which of course may not always be known right away from early feasibility, but in each case it will help us define what real formulation be and what process then associated with that to ensure the product itself will end up the right way. So some general formulation approaches. We've established a target product profile. We're in the design phase here. What are we looking at here? So we have maybe something that we could do a neat formulation with just buffering components, and that's because we need a high dose for some reason. And really just keeping things as simple as possible and understanding what is the stresses on the active itself. A lot of times it's a very realistic option. Proteins themselves in the dry states are relatively stable, and as long as there's no impact on the surface of the particle, so we can process these. And again, we like to kind of keep things as simple as possible. Why add complexity if you don't have to? In a lot of situations, though, you do need some type of stabilization. So we do that. We can add excipients to help with the active itself. In some situations, we need a bulking matrix based on the fact that maybe you don't need such a high dose. You're just trying to put something in the dry form that you can use for the target delivery profile. Maybe we know we need to have improved powder handling, so we can add certain excipients to impact surface interactions that will improve handling. Then, of course, performance, which is a big thing across the board. This is, we can introduce densifying agents, surface acting agents, modulation of particle characteristics. In some situations, you can make things that are a little more porous, dimpled, or spherical, things like that. And a lot of times, the excipients themselves will help define what that rate will be. And one thing I want to note as well is so macromolecules themselves during the spray drying process typically do exhibit kind of a slow diffusion step because we're using a water matrix. So when that happens, you have the protein itself that goes and enriches the surface of the particle upon drying. And so it does lend itself to potential issues on the air-liquid interface when spray drying. So a lot of times we'll include excipients that will help protect the macromolecule itself away from the surface while drying. This is kind of a distinctly different thing than lyophilization when you're worried about cake formation. Excipients themselves are a little. So big thing as well, so we want to actually formulate around a primary failure mechanism that is typically aggregation. So we see this a lot across different protein formulations, and the aggregation itself is sometimes really hard to characterize, but really has a big impact on where the product itself can go. So I'm just kind of showing a general mechanism itself on this slide. And again, a lot of the times the introduction of the shear stress through the atomization process can change the overall secondary structure of a molecule and therefore lead to an aggregation potential. And again, it has a lot of downstream impacts on product development. So if we do know how a molecule itself will aggregate initially, we can then look at formulating that. But again, that's not always known, and so there's a kind of a platform approach where you can use for formulations to make sure that you're not going to have this introduction within the process development. And a lot of the times, too, aggregation is a function of its surrounding environment, its pH, different ionic strengths, and so we're looking at things that will help provide that protection throughout the process. Through the next couple slides, what we'll do is we'll discuss what excipients can help actually drive or decrease the amount of aggregation within the product development steps. So the big thing as well is when actually converting a macromolecule to the dry state is its thermodynamic instability. So I'm showing just a basic model here around that instability. The first way really to control the molecule itself, which includes aggregation we just touched on, is the control of the dehydration step during drying. So a basic approach to this is including hydrogen bonding sugars, things like trehalose, sucrose, even glucose in some situations, and those sugars will directly interact with the molecule and replace water during the drying step. In a lot of situations, if you do not include these sugars, as we would expect even in a liquid form or a lyophilization or something like that, you'll lead to an unstable secondary structure associated because of the stress that's been put in the situations. And kind of said before, sugars are able to help insulate the protein-protein interaction upon the drying step. So I wanted to break down here in this slide really are what are the specific rationales for the biopharmaceutical formulations and what excipients that we look at in each case when looking at feasibility studies. So, the rationale, as I said before, maybe a bulking agent. You just need something for dilution steps. So something that's an inert that won't pick up water. Typically, an easy way of doing this is adding something like glycines, cysteine, proline, just a basic amino acid. Now, if we need actual stabilization during, let's say, the engineering process, we need to protect the protein from the air-liquid interface, we use a lot of times surfactants, and these are polysorbate 20, 80, a poloxamer, or something like that nature. And as we talked before, now these API stabilization in the solid state, so this replacing of the water or the dehydration step, we need to add things like trehalose or maybe even dextran, just a sugar polymer, in a lot of cases what we're adding. We'll also look at adding amino acids such as histidine or proline or arginine to help in the buffers when processing the materials. And in special cases, we have added EDTA, sodium chloride, calcium chloride, things like that. Again, the focus there is to have protection of the active while we're processing through spray drying. And then we'll add other components as well to kind of decrease the amount of particle agglomeration. So again, it could be the same excipients, but they have different rationales or inclusions. So trehalose, a big one here you'll see in the slide that's used. And the actual ratio of trehalose is important, and we'll talk about that a little bit later in a case study at the end of this webinar. And then for particle dispersibility, you're looking at things that will help protect the surface or form a shell while spray drying. So you have surfactants. A big one here is actually leucine. And then for lung application is the phosphatidylcholines. And if you're focusing on oral delivery, which we'll touch on the next slide as well, a lot of times you need an adjuvant or a mucoadhesive agent to help kind of stick that specific API in its site of action. So examples of this are chitosan, amylopectin, a caspase, capric acid, things like that. So there are formulations specific more now to delivery route. So 4 main delivery routes that we work with are inhalation, intranasal, oral, or potentially an IV sub-Q delivery. So now inhalation, the primary goal there a lot of times is particle size. And so you're looking at engineering a particle that will fly and disperse and have the right association in the lung. And so you have bulking agents, again, for stabilization, such as trehalose. All the times you're using salt for surface protection or even API stabilization, citrates, chloride, or surface modifier, and again, which is leucine or the phosphatidylcholine. Intranasal, very similar to inhalation as a particle size main focus, but you can add a couple different excipients here. These are usually polymer-based to help protect, control the actual dissolution rate of the product itself within the nasal cavity. Now for oral delivery, which we'll highlight on the next slide, there's a couple different excipients you need to now include to protect the macromolecule and also include during spray drying itself. You have polymers for protection, things now that need to also allow for release within the GI tract and protect from the gastric media, which is low pH. And then lastly, for IV sub-Q, you're looking at things, again, bulking agents, but the big thing here is sterility. So you need to focus on how the process itself is controlled while spray drying more than the formulation components, because the attributes that we would use for there are the same things we would use for the other delivery modalities, but we just need to have something that will be fast dissolving. Okay. So I wanted to highlight briefly on kind of the oral delivery problem statement. So in this case, we had a target molecule, a peptide, that had relatively high aqueous solubility in this case, but was very pH sensitive. And we had kind of known stability issues at room temperature. So path forward in this case was including something of an active loading 50%. This was kind of hit the high dose, but because we had some instabilities, we knew that we had to add kind of a high TG matrix to stabilize the molecule and control dehydration, and then also have the associated buffer salts to make sure the protein itself was not unstable during the process. So we added dextran or the PBS salts. This, in the end, would also provide kind of a fast dissolution of the target site. We included a surfactant, in this case, poloxamer, to give protection to the particle interface while spray drying. And then we included a permeation enhancer, capric acid, to help improve absorption. Now, because it's an oral delivery platform, there's a dosage form of this association. So, okay, we selected a formulation, we screened it, we kind of selected a baseline spray drying process, but now it's a matter of we collected those spray dry powders, we have to compress them into a mini tablet and then coat it with Oidrogate itself to get your defined pH release. And there's kind of this model here on the bottom to kind of go through that process train. And analytically, we looked at, okay, what's the release and the overall potency of the active itself of the different pH characteristics. So we've touched a little bit on formulation itself, and of course, formulation, we want it to be as basic as possible, includes things that will stabilize, and each molecule kind of sometimes depends on different ratios. Sometimes if it has a lot of hydrogens, we need to stabilize these, we'll add more sugar. Maybe we need to add amino acids that will help solubilization or control ionic charge, things like that. But, you know, there's a wide range of things we'll look and investigate and design around. Those implications of the formulations have a big impact on the process itself. And so We can design a formulation, but we need to see how it's going to behave to make sure we are actually creating the right product in the end. So as I stated before, the process is broken up into 4 different sections. We have a solution, an atomization step, a drying step, and a final collection. Now, solution itself is really all about kind of what the API can handle and prefer. So what's the overall pH? What kind of solids concentration can we push? And that's really, again, an impact on throughput, but also impacts on how atomization stress may be or the drying kinetics that we'll see as a function of the solids content. And then also what kind of temperature profile. Sometimes these ranges can go from cold. We could hold that in there in refrigerated conditions to ensure that we don't have any instabilities. Or in some cases, an elevated temperature may allow us to include different excipients and give you different kind of product profile. So the solution is fed into the atomizer. The atomizer puts on shear stress, and there may be a certain molecule itself that has an issues with the shear, and so we have to make sure that that formulation is providing the necessary protection across the board. And then given the solids content, atomization is tailored to give you the desired particle size. So we can use different atomization pressures, low PSI to high PSIs. We can change the nozzle size to form different droplets. Again, this is a function to ensure that the active itself is stable and within the droplet, and then will lead to a dried component. The drying is the final relationship between kind of the solubility in a solute and then the excipients or materials included. So let's say we added certain excipients that have very slow diffusional rates. That may impact what our process conditions are going to be. And we have components that are fast drying that will flash to the surface. Again, those considerations need to be taken into account when designing your process. And again, as I said before too, the protein typically diffuses slowly while drying, so the material can be located on the surface. And so if we want to add different excipients, all this kind of balance, again, is on the overall solubility and association of the protein itself. In a lot of cases, too, what we can do is add something like leucine, which will give you crystalline character. So it's a fast precipitation on the surface, protects the materials away from the surface, and also can control the water sorption profile. So anything that's usually fast drying will be spherical or hollow, and slow drying materials are usually wrinkled and be a little bit more dense. So lastly, the collection itself by the cyclone is also critical. In this case, it really comes into account what is the overall product yield. So by having the right initial input and drying kinetics, and then having a high efficiency cyclone that's able to separate the dense particles from the inert gas, we can have a nice high product yield, which is typically greater than 90%. This question comes up a lot of the times too when considering the process and the formulation. So, what kind of temperature profiles can we do? So I have an inlet temperature that a lot of the times is relatively high because it's defining the overall thermal constraints within the spray dryer. So what we provided here on the right-hand side of this slide is our kind of model of typical profiles and operating space. And again, since we're utilizing evaporative cooling, which focuses more on a wet bulb temperature of the selected solvent and a fast drying step, we can push the high inlet temperature a lot because the product just doesn't see this. And these inlet temperatures can really range from anywhere from 120 to 160 degrees Celsius. And usually what defines our high end of our outlet temperature is based on the equipment constraints of what the hot gas can get to. And in the end, the relationship is all about the liquid to solid phase transition and not necessarily the thermal profile that you're seeing within the spray dryer inlet that's been selected. And just kind of a brief representation, the bottom part there too, we did a hot bench experiment to understand how the material itself would be able to handle temperatures. And then also for spray dryer outlet temperature. Now, this is actually the critical parameter when understanding spray drying and formulation components that are added in. So the inlet, as we just talked about, can set up the actual thermal profile, and then also in combination with how insulated the dry chamber is, But those inputs now give you the output of the outlet temperature. And we are really concerned a lot of times with what that outlet is. And so, we're looking at outlets that will range from anywhere from 40 degrees Celsius to 80 degrees Celsius. And can that product actually handle that? Because it will heat up to that outlet for a brief period of time prior to moving on to the collection vessel, which is usually sort of in between a room temperature and that outlet condition. So we're balancing this thermal constraint within the spray dryer and the evaporation kinetics to the overall product profile of the molecule itself. What we did on a quick study here on the bottom left was tracking impurity growth of a specific peptide at different outlet temperatures and times. So now, again, those times, usually the product itself won't see a 4-hour, but there's sort of an extraneous condition, so we can really see if there's a drop-off in overall impurity profile, which we did see that in this specific molecule, after an extended period of time. But it wasn't really a function of temperature. It was more of just a time issue at that elevated temperature. In this case, anything from 50, 70, or 90 C. So if we know that we're going to have some instabilities past this 50 C, that's sort of going to be our constraint within the process itself. And then one last consideration with process and formulation itself as well is the relative humidity and corresponding water content sorption profile of a product. So, a lot of times we need to be very, very strict about what the water content needs to be, because we'll know that there'll be some instability associated with it. So we do define kind of what the overall outlet percent RH will be, and that's given by the operating condition. It can be highly tunable and usually highly robust as well. And we will do a kind of a dynamic vapor sorption profile to help identify water content as a function of relative humidity to help to design the initial safe space. So I wanted to highlight, too, the fact that spray drying itself is, by nature tunable, and everything we do in early formulation screening can move along to preclinical process development and clinical commercial manufacturing. So you can see the images on the slide here, which show 3 of our spray dryers in-house. We do have these in development space and also, as I said, in kind of a GMP space. So anything, again, that we identify during that lab scale process can be moved along more or less by simple kind of mass transfer models and things like that for product development. And again, one thing to add is to help throughput, a lot of the times you can use multi-headed nozzles. It really depends a little bit how the material itself dries, but can be included. And then we can also utilize kind of optimized single cyclones to help increase collection. We've also identified a couple different specialized equipment designs to help enable droplet drying. So because you're using a water as a matrix, sometimes these materials slowly diffuse, and if you do not have the necessary drying time within the dryer, you'll get impaction on the side walls of the spray dryer or even the pipework prior to the collection vessels themselves. So we've designed in-house some extensions of these spray dryers, either on our lab scale or on our what is known as a PSD1, sort of large-scale manufacturer. Again, just gives the ability for the particle itself to dry longer, leads to better particle collection and overall morphology. And really, it's going to be specific to the product itself as well and the excipients that are included. So now, of course, with any product screen, analytical is critical to understanding what you're actually engineering. I'm not really going to go into specifics, about each one of these analytical techniques. This isn't really novel. It's obviously in the literature. A lot of folks use all these different techniques. I just wanted to state that we do use each one of these a lot of the times when doing our characterization, whether that's the solid state or even some wet lab analytics to ensure that the molecule itself is stable and processed correctly. And in the end, all we care about is hitting the product profile, making sure the molecule itself is stable and usable for downstream processing. So just in the last couple minutes of this webinar, I wanted to touch base on a brief case study that we did here, kind of give you, again, another idea of what we would do formulation and process-wise. So the program goal itself here was identify the best formulation and process space to help deliver just a dry powder that would reconstitute at a higher concentration. So we're designing formulations really to have just kind of a short or intermediate stability. This is a kind of a bulk product itself, but just trying to push how that monoclonal antibody can resuspend. So for our experimental approach here, what we did is we designed formulations that varied active loading, sugar content, a surfactant type. The actual buffer salts identified were really based on the active and its pH and things like that. So we didn't add any of those in the formulation screen. So you can kind of see that table in the bottom part of the slide. And really more of a mini DOE to understand what is the relationship between the active itself and the components and the process. The process, we focus more on atomization, so what kind of pressure and shear stress we put in on the product, the droplet itself. And we also added a solvent difference here. So we added ethanol. In this case, we were trying to make particles that would dry a little bit faster, have different morphologies that may actually then impact how material would reconstitute. And we did also look at the temperature outlet. So the powders were collected. We assessed them by water content. We did morphology. We did reconstitution quality of that elevated condition, and then also looked at aggregation by SEC. Process yields, you can see on this slide as well, varied a little bit. And we'll say that kind of the product yield was more of a function of actually inclusion of ethanol in this case, dropped it down quite a bit. We might have been actually making smaller particles. But all in all, high yields on most of the products greater than 90%. Water content was a function of the excipients as well, but in between 0.5 and 1.5 weight percent. So in this slide here, we have the reconstitution profiles. So we did see impacts from the formulation and process itself on reconstitution. So, you know, on the graph in the bottom here, the native solution itself, so non-spray dried, 20 mg per mil, had a relatively low turbidity count. We tried to push the concentration 5x, and then had kind of a target profile of 30 NTUs. A couple of these formulations certainly did fail that specification as a function of a couple of different including components and process conditions. While there was also a number of different formulations here listed as A2, B2, and C2, which had kind of varied active loading, atomization changes, and surfactant inclusion, which were almost near the same turbidity or particulate counts within the formulation itself, which is a benefit in this case, right? So we were able to push reconstitution 5-fold by using the spray drying and identifying a formulation going forward. So each one of those solutions themselves we're looked to see now. So why were we getting impacts on turbidity or particulate counts? Ended up not actually being associated with aggregation, so by SEC. So these were sort of almost soluble aggregates or soluble materials within the solution that were giving us particulates and not associated with protein instabilities. So sort of kind of an interesting findings here in this case. So just to highlight this, you know, of course, in the case itself, formulation process did impact the properties when it came to reconstitution. Inclusion of the sugar and surfactant dramatically improved product performance. But when we increased the sugar ratio above the 300:1, that's a molar ratio, we had negative response when it comes to particulate counts. The surfactant type did matter. We screened, in this case, the polysorbate 20 or polysorbate 80. And a lot of situations, it really can be important. Now, we did change as well the sugar inclusion, so either mannitol or trehalose. It's found out that trehalose did a better job in the formation, the particles themselves, and reconstitution. And atomization pressure impacted particle reconstitution. So in this case, a lower atomization or lower shear stress helped our particle reconstitution. We're going to continue to move this study forward and kind of look at some further studies around adding an excipient to help decrease viscosity, which is something like arginine, an arginine salt. Try to push limits of process throughput and then increasing flow rate. So now, in the next 2 slides, we'll essentially try and conclude this webinar. So I know there's a lot of information with it that we've gone through. Obviously, there's discussions around formulation, process. In each case, a lot of the times that is specific to the molecule itself, but we do have a general platform approach of what we would include and screen on the front side so you're not wasting a bunch of time doing big, massive DOEs. In each case, though, we want to take the holistic approach of really understanding what does the product needs, what kind of structure, function do we have. Is this going to be an intermediate profile, direct delivery of the material? Do we need high reconstitution? Can it be low reconstitution? We bulky matrix, things like that. So we're understanding what the product needs, then pushing that to a formulation to help support those product needs. Add only excipients that are necessary. Let's not add things just because. We want to make sure they have a functional component within each one of the particles to help stabilize and improve performance. And then tailor the process around these individual formulations that we've included and then the product itself and its profile. So if you can take into each one of these into account, really, we'll be in sync and have a nice, robust path forward for formulation and process development early on and down the road. So I just want to conclude this webinar now and really just state that spray drying itself is precedented, it's robust. You can see it's tunable. We talked about how it's scalable. It really can be an enabling technology for biotherapeutics. There's a lot of different agents you can add to help stabilize, but there's a couple basic ones that we've touched on already. And again, what you really want to do is just focus on stabilization of the molecule itself. So what does it require to make sure that it has the same secondary structure upon processing and drawing, and then also is able to hit whether the receptor or whatever function it needs to be in its delivery profile. Or in the cases of bulk stability, and you're just trying to do an intermediate processing, have all the necessary excipients in there to keep it nice and stable in, let's say, a room temperature, elevated temperature, and you've densified the particle and you've decreased the footprint. So with that, I just want to thank you again for listening to today's webinar. I really hope that the information provided was useful for you and kind of spark additional discussions.
Thank you very much, and we're going to move into an interactive poll question here. Please go ahead and select your answer and hit submit. So this question is, would you like Capsugel to follow up with you after the webinar? That's, would you like Capsugel to follow up with you after the webinar? Yes or no? Please go ahead and select your answer and hit submit. I'm going to leave that on screen for a couple of minutes, and while we do that, we'll go ahead and get started with our questions and answers session. So people have been sending in questions all throughout this webinar, which is brilliant. Please do keep them coming in. Just a reminder, if you would like to ask a question, you can do so by typing it into the box at the top left-hand corner of your screen and clicking submit. So we're going to go with this one first. This question is, with a water content rate of 1 to 7%, are you concerned with stability of the protein on stability?
So to answer that question, I'd say yes and no. I mean, it's a little bit of function of what is the actual degradation profile of the molecule itself. We've certainly processed materials that have had high water content. In certain cases, dehydrate further, you lead to instability, so the water helps insulate a little bit of that material. But, I think it's a range. In most cases, we are concerned with water content and long-term stability, so we'd like to keep it as low as possible.
Okay, lovely. Thank you very much for that answer, Michael. And I'm just going to leave the poll up just for a little bit longer while we take one more question. This question is, are biologics drugs sensitive to heat exposure?
Yeah, I would certainly say a lot of biologics are susceptible to heat exposure. So you're looking at things that either will help denature or unfold the protein in solution. So you can do a lot of the front side stuff at looking at solution DSC. And so let's say identifying the right buffer composition and you see an unstable molecule at 60 degrees Celsius, then you can design around your process to make sure that you're never going to reach that temperature. And again, the spray drying itself is relatively gentle, and you're not really concerned about your input or your inlet temperature, it's your output. So it's a function of really just moving excipients in that will help either increase the overall glass transition temperature, which will come into a combination with the molecule itself, and then give you a higher stability, or if the susceptibility of a neat formulation, you're just going to try and stay away from what that thermal profile is. But yes, in general, the biologics are certainly, as like any other entity, susceptible to heat exposure.
Lovely, thank you very much. And I have now closed the poll. Thank you to everyone who voted in that. Moving into the next question now, this person would like to know, are whole cells still viable if they undergo spray drying?
It's a good question. And so we have in the past looked at spray drying whole cells, and they have certainly stayed intact and been able to kind of hit their target site. It's a little bit of a balance here in this case as well. So that's one of those formulations that usually you actually don't want to dehydrate too much because you're going to have a lot of residual water within the cells. So if you use an aggressive drying condition, it can lead to lysis of those cells. And so you're going to want to protect gentle processing conditions, low temperatures, low kind of flow rates, low shear stress. Add excipients again, in this case to help kind of bulk the particles themselves so that you don't have the cells on the surface of the particle. So you can simply do that by adding kind of inert sugars and also additional buffer salts is kind of the generic approach for that.
Okay, lovely. We're going to go straight into the next question now. Do you take additional precautions when cleaning post-spray drying of biologics to prevent cross-contamination?
Yeah, I mean, I think that's a good question too. Obviously, there's a lot of process controls to this. Effective cleaning between agents is important. A lot of the times though, you can just use kind of high pH, so basic cleaning solutions that will allow to flushing the sides of the stainless steel dryer. But in between, I'd say runs, especially when you're doing, let's say, an early feasibility study, you want to clean in between the spray dryer because you don't want to have any carryover, and there can be carryover in between each spray. So I would say certainly yes, you take extra precautions in making sure that you're not having residuals or things like that within the spray dryer and impacting the integrity of your product.
Okay, lovely. Just enough time for maybe one or two more here before we wrap up. This question is, what is the main value proposition for spray drying proteins? Are there any commercial biological products that use this approach?
Yeah, so I mean the main, I'd say, benefit of using spray drying for biologics is the idea of particle engineering. So you can add different excipients, you can control morphology, you can control dissolution, you can increase stability because then you're also putting things in to help interact with the protein. So it's really just as making that an ingoing biologic a better product, as opposed to other technology, shall we say. So the second component of that question, actually, Tara, can you just repeat that? Yeah,
I can. The question was, are there any commercial biological products that use this approach?
Yeah, so there are a couple different commercial products that have sort of used this kind of spray drying process. You have, let's say, a number of inhalation biologics. So inhalable insulin was a big one that has kind of promoted the spray drying process. And there's certainly a number of products as well that are kind of, let's say, in the clinic that are utilizing this as a path forward.
Okay, lovely. Just enough time for one more question here. And the question is secondary drying necessary for spray-dried biologics?
I would say to answer that, it's not necessary. and usually because what you can do is you can tailor the process itself with its thermal profile to ensure that you hit the water content that you want or desire for your product. But secondary drying can also be implemented. The problem with secondary drying really is now controlling either sterility or bioburden or microbial growth itself, because you're adding an additional unit op. So we'd like to stay away from that if at all possible, and just use the process to define what your actual content, your residual solvent content is in the product.
Okay, brilliant. Thank you very much for that answer, Michael. And thank you to everybody who submitted a question today. Unfortunately, we have received a lot more than we have time for today. So those are going to be, the remaining questions are going to be answered offline. So you can look forward to a response there. But that just leaves me to thank Michael for what was a great presentation. I'm sure you'll all agree. And to thank Capsugel for sponsoring this session. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar if you'd like to watch it again, or you can access it through our website. The website is www.business-review-webinars.com. And once this webinar ends, a survey will appear in its place. We would appreciate it if you could stay there and answer the questions for us. Your feedback is important. We do look forward to sharing further webinars with you. So do keep an eye out on our website, just mentioned. Follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group as well, Business Review Webinars. Thank you all once again, and I hope you all have a lovely day.